I'll hand over to our host. We're very lucky today to have Marie Boran, science journalist and regular contributor to the Irish Times, to introduce our event and take us through the programme. Thanks, Marie.
Thank you, Richard. So before I do start, I just do want to remind people, if any questions occur to you throughout the talk, just pop them into the chat and we'll have a look and try to get to them at the end. But yeah, I'll get right to it then. Good afternoon and welcome to the inaugural webcast from NeuroInsight to mark the launch of this new research programme, which is led by the Royal College of Surgeons in Ireland, and it's working across the National Future Neuro SFI Research Centre for Chronic and Rare Neurological Diseases and the Insight SFI Research Centre for Data Analytics. And today's webcast, which is fittingly happening during SFI Science Week, which is great, it's on the topic of new frontiers at the intersection of data science and neurology. And I say fittingly because this year's theme for Science Week is actually creating our future, a national conversation between the general public, the research community and policymakers. And I guess this is part of this conversation, isn't it? We're doing a webcast now where all kinds of publics, expert and non-expert, to get to actually get a glimpse of the kind of research that we will hope to be seeing happening in future neuro and NeuroInsight and in Insight in the future. I've said future lots, but anyway, I'll get on to it. So what are these new frontiers, I hear you ask? Well, I'll just give you a little background information first. Our scientists have obviously made great leaps in developing AI over the last few decades, and machine learning and programming that can process data like humans and analyze complex multi-format information streams even better than we humans can. And today what we want to do is explore how these thinking machines, or AI and machine learning algorithms, actually can help us with difficult challenges in human health. And here in Ireland, just to give you some background information and statistics, 1.1 million individuals, or 25% of the population, are actually living with some kind of neurological condition, ranging from relatively minor disorders to serious chronic and rare diseases. And this lunchtime, we've got a series of distinguished researchers that will each give a lightning talk inspired by this question: how can AI, machine learning and intelligent algorithms be used to understand, detect and find new ways to diagnose, treat and manage these conditions? And so today's meant to inspire conversation and ideas around this programme, not just amongst researchers and collaborators in companies and hospitals, but also importantly patient groups and the general public. So before I actually introduce our fantastic lineup of speakers, what I would like to do is introduce NeuroInsight Programme Manager at Future Neuro, Richard Tuhig, who's going to say a few words about the research programme before I kick off.
Thanks, Marie. That's brilliant. So NeuroInsight is a new research funding programme, and it's on the Murray Sklodowska-Curie Actions Model, the MSCA. So if you don't know those, they're like the European Union's high potential grad scheme for researchers. So they identify the greatest talent amongst early career researchers, and they give them funding and time to develop both their research capabilities, but also their professional skills. So they're training up to be the next generation of research leaders. So we have funding from the European Union. It's matched by funding from Science Foundation Ireland. We have enough money to fund 33 people, 33 two-year fellowships over the next 5 years. And the way these work is we invite people from around the world to submit proposals for research to come and do based in Ireland. So mobility is important. We still think mobility is important for research careers, that people move around between countries and labs and cultures. So we're inviting proposals for research on the themes of Future Neuro, which is challenges in neurological disorders, and Insight, which is challenges in data analytics. And we're looking for research that links the two. So, I mean, you could imagine that would be chemistry, medicine, biology, data science, but also you can think about questions around ethics, around sociology, economics, even the creative arts, and we're really open to all ideas here. If you want to apply or want to know more about the programme, go and look at our website. That's where all the details are. And if you want to steer the research programme towards a particular direction, now's the time to get in touch. And so today, we don't have any researchers here yet. We don't have anybody in place, but we have challenged our existing research leaders to come up with directions where this program should take us or may take us. And I'll let Marie, you can get on with taking us through that. Thanks.
Great, that's excellent. So before I actually go into introducing the first speaker, I just want to go and just briefly list all 6 speakers so that you kind of see what we've teed up for this afternoon. So first off, we have Alan Smeaton talking about service dogs, and you'll probably I won't give that away until I introduce it, because you're wondering why is he talking about service dogs. We'll get to it. Then we have Corrigan. He's going to talk about data analytics and electronic health records, which are obviously very topical these days. And next off, we have Claire Gormley talking about advanced statistical methods, and Deirdre Kilbane is going to follow her. She's going to talk about AI and how it's applied to RNA sequencing. You probably go, oh, RNA, mRNA. I've heard all about that over the last few years, what with COVID. And we've Tomas Ward from DCU, and he's looking at using AI outside the clinic to understand and treat neurological conditions. And finally, we have Gian Piero Cavallari, and he's going to talk about AI as a tool for faster, more accurate genetic diagnosis. And without further ado, I'll get straight on to Professor Alan Smeaton. Alan, welcome. Thanks for being here today. A little background on Alan. He's Professor of Computing at Dublin City University, and he's a founding director of the Insight SFI Research Centre for Data Analytics. And Alan's expertise is in analysis, indexing, and retrieval of all kinds of visual data, with a particular focus on video. And some accolades that I'll mention if Alan doesn't mind. He's a member of the Royal Irish Society, a winner of the Academy's Gold Medal for Engineering Sciences in 2015, and a former board member of the Irish Research Council, and a fellow of the IEEE. We could go on listing lots more, but sure, we'll get into Alan's talk instead.
Yeah.
So Alan's going to talk about wearables, service dogs, and seizures.
Thanks very much, Marie. Good afternoon, everybody. That intro didn't form part of my 3 minutes, right?
Yeah, I know it's a bit long, sorry.
So there are almost 40,000 people in Ireland with epilepsy of some form. And one of the things that we can do to help them is look at how assistance dogs can help them. So anybody who works in the area of epilepsy, or who knows anybody who has epileptic seizures, or who has seen this, will know that it's a very, very traumatic event, or can be a very traumatic event. And what we can do is we can use assistance dogs, as the video just showed there. Maybe if you go back and show it, play it again, then we can use assistance dogs to train to help to limit the damage or self-harm that can happen when somebody is having a fit. Now, this is a training example here, where the lady on the floor is simulating a seizure. And the lovely looking American pit bull terrier there has been trained to basically insert himself between the lady's head and the ground. Now he takes the damage, he takes the hit, and that's great. And there are other kind of things we can do with assistance dogs to help during an epileptic seizure. However, if we could detect the onset of a seizure, then the person could place herself or himself in a safer place, awaiting the onset or possible onset of a seizure. And service dogs or assistance dogs are known to be able to detect the seizure onset, probably via their scent and their ability to detect volatile organic compounds. So let's see an example of how this might work. If we go to the next slide there, Richard. So James is an epileptic. He gets seizures on a regular basis. He has, of course, no recall of when these happen. And when he gets some seizures, what has happened is that he has taken the shirt that he is currently wearing, which he wore for his previous epileptic seizures. He has taken that and he has frozen it. So any VOCs which he emitted at the onset of his previous are now captured and embedded in the fabric of the no, don't hit it yet, embedded in the fabric of his jumper. So he's frozen that and put it in the freezer. He's now taken it out. I'm sure he's thawed it a bit, and he is now wearing it. Ralph, who'll be introduced in a moment, is a dog who doesn't live in the same house as James, but Ralph is a black Labrador and has been trained to detect the VOCs, which are indicative of the onset of a seizure. And when he detects this, his training has instructed him to press a big red button. Okay, let's see what happens. If you play the video now, Richard, you see. So what happens is that, just click on it. Just click on the video and it should play. Now, and nothing I can do. Just click on the video and it'll play, Richard, anywhere on the screen. This did, of course, work beforehand. I'm not seeing your mouse moving, Richard. If you click on the video, on the frame of the video, it'll play. Anyway, I'll bluff while what happens is that Ralph, the Black Labrador, is introduced. He goes over to James, gives him a big licky face on, you know, as dogs always do, big lick on the face, then smells his jumper. We're going back to screen shot. I'll carry on. Then we'll smell his jumper, we'll detect the volatile organic compounds, and we'll then look for the red button, which happens to be in the hand of Katie, his partner. So we'll go detect it, press the red button. He'll go back to James, smell again, detect, and press the red button. And then what happens is that Katie gives the dog a bit of a reward, and then moves to the other side of the room and puts the red button up on a shelf, which is totally out of the reach of Ralph the dog. So this kind of works well in cases where the subject, who could have an epileptic onset, is in a home environment or in an office environment where the button is always in the same place, where the dog knows where the button is and will always go to the same place. But what if James is not at home and not at workplace, because he just doesn't live at home and workplace? What if he's in a supermarket, or he's in a restaurant, or he's in a cafe, or he's out for a walk somewhere, and he exudes VOCs, the dog detects, and the dog is looking around for the red button? What can we do, etc.? What if the service dog instead was wearing wearables, which could automatically, which could automatically pre-detect the onset of the VOC? Previous slide, Richard. So in that case, what would happen is that a dog with a wearable, now play that video. Yeah, thanks. Okay, so there we are, James. Dog comes in, big licky greeting on the face. Oh, you've got that VOC, smelly stuff. Where's the red button? I'll press the red button, which is Katie's hand. Let me see. Oh, more VOCs. I'll go and press the red button. There you go. And then Katie will go and put it out of his reach, which is completely defeats the purpose. Now let's go to the next slide, please, Richard. We go to the next slide. The button can be connected to an alerting system, but if he is not in the home or not in the office environment and he's out for a walk, what if the service dog had a wearable, a simple accelerometer around the collar, which could detect a particular kind of revealing behavior that the dog was trained to do, such as a dog spinning. So instead of looking for the red button, the dog just spins. That is much more easily to detect automatically and would work in a restaurant, cafe, out for a walk, or in a shopping center, or whatever it would be. Next slide, please, Richard. So what I'm interested in is doing work at the confluence between epilepsy and patient needs, anticipating the onset of a seizure. I'm interested in that and how that intersects with animal behavior science to detect onset and then to notify in some behavioral obvious way, such as pressing a red button, not so good, or doing spins, very good. And to enable that, wearable sensors and machine learning to detect kinds of behavior. So there's a huge opportunity around assistance dogs and detecting seizure onsets. And this worked example of a spinning dog, which happens to be my dog, by the way, not just doing it for tricks, is just one possibility in that space. That's it. Thank you.
Alan, thank you very much for that talk. It's really interesting. I'm just wondering, going forward, how do you see this being something that might make its way to the supermarket shelf, even, that it would be something that anybody could buy? Because they actually seem like they're all existing. Like you said, this is the confluence. These all separately exist as things that are, you know, like existing and working. So do you see it?
Yeah, joining them together. Well, I think once we know that people do exude VOCs at the onset of an epileptic seizure, and we have a dog that can detect that, what this is doing is it's filling the gap and joining those together, so that we have wearables that we use, such as wristwatches and Fitbits and rings, which can detect minutiate details of our behavior, whether we're standing, sitting, walking, lying, running, jumping, all of that sort of stuff. So it should be easy to then reapply that to service dogs, provided there's a consistent action that they indicate. Not a red button, but a spin or a rollover or a whatever it might be. And that's where the animal behavior scientists come in.
Fantastic. And you can imagine that data building up and being able to share it with your GP too. So great stuff. Thank you very much, Alan. And we're going to move on to our next speaker, Dr. Derek Corrigan. So Derek is a postdoc researcher at RCSI, and he's part of the Future Neuro Centre. And he's both an experienced researcher, but he's also an industry leader, having been a software developer, management consultant, research manager for the ICT, health, telecoms, financial, and public sectors. So there we've got Derek's great. He's a man wearing many hats. His doctoral studies were on developing electronic and computable ontology models for clinical prediction rules populated from primary care data. And today, Derek's talk is going to be on developing the epilepsy electronic health record and here in Ireland, which is going to be quite interesting to hear all about this. And that's a growing area now, having these subsets of electronic health records for people with different kinds of chronic diseases. So Derek, I will let you take the stage.
Brilliant, many thanks, Marie. Okay, so as just said there, I want to talk about a vision for the development of an epilepsy learning health system. So by way of some context to this presentation, I suppose at the centre of this is the National Epilepsy Electronic Health Record. So this has been in operation in Ireland since 2009. There's nearly 10,000 epilepsy patients contained in that record. It's one of the most mature electronic health records in Ireland and has really been a valuable resource in terms of clinical care in outpatient epilepsy clinics across hospitals in Ireland. But obviously it's a really rich potential source of analytics data that could be used for service improvement. So we see our vision of analytics as very much two sides of a learning health system loop. One is analytics to provide evidence generation, to generate things like predictive models, looking at things like seizure response over time, what factors may be associated with that we can delve into the analytics and see those sorts of associations, and looking at population-based analytics as well, understanding the population that we're treating and are we reaching the right people. So evidence generation is one side of that, but also we're very interested in this feedback loop of knowledge translation. So how do we take that evidence and feed it back into actual clinical practice and see how we actually impact on clinical care and improve clinical care? So that aspect is often missing in a lot of analytic solutions. So next slide, please, Richard. So in terms of, I suppose, progress to date, I've been working in this the last 2 years. We've looked at a number of analytics areas that sort of build collectively on top of each other. So we've looked at the area of operational analytics, where we look at the characteristics of particular patients in our service. We've looked at medication safety reporting around use of drugs like valproate, for example, in terms of treating people with epilepsy. We've also looked at COVID performance of our services, both sort of 3 months pre and post the lockdown, the first initial lockdown that happened, and the use of telecare and how that impacted on service delivery. That was picked up as well by the ESRI in a report in terms of those analytics. We've also then looked at research-driven analytics, more where we've got research hypotheses. We implemented the EpiDive project, which looked at over 6,000 patients and the characteristics of patients taking anti-epileptic drugs and how those related to seizure response in those patients. We then sort of have dipped our toes into the area of personal analytics in terms of development of prediction models, looking at development of regression models, for example, proof of concept models in terms of looking at factors that might be associated with drug-resistant epilepsy in patients. So these are all the areas that we've sort of worked on to date and that we wanted to develop into this learning health system. So next slide, please, Richard. So I suppose there's a lot of challenges that we're looking to address and maybe work with people who are interested in collaborating with us, and a lot of opportunities and solutions that we see in addressing those. In terms of challenges, obviously because we're working with healthcare data, this data is not our data. We don't own this. It's owned by the patients and the joint data controllers in the hospitals. So there's very strict, obviously, ethical guidelines in terms of how we can do this sort of analytics. And we apply for ethics applications to the hospitals and do full data privacy impact assessments. But that process, while it's necessary and absolutely really important, there's a long lead time on that. It takes a long time to navigate that. So in terms of that represents a challenge for predictive analytics, where getting access to data is obviously the lifeblood of doing predictive analytics. And you often think then of perhaps analytic studies that you might have wanted to do, but you didn't think of during the initial ethics application, so you can't do them. So there's I suppose a roadblock there in terms of getting access to data, but it's necessary to protect patient privacy. Obviously, measuring clinical impact of analytics is really challenging. I mentioned that feedback loop in terms of translating knowledge back into clinical practice, and that requires development of actual tools to feed that data back into frontline clinical practice and see, does it actually change clinical care? But there are solutions that we see. We've dipped into the areas of looking at synthetic data generation, whereby you use machine learning methods to generate data sets that have the characteristics of the population as a whole, but are synthetically generated so they preserve patient privacy. But we can still use that synthetic data. We would see it as a vision for doing rapid prototyping of models, moving it forward until we can get all our ethics approvals processes completed and can apply them in practice then to real data, but it still allows us to proceed on that basis. And that allows us also then potentially as well to do app development in terms of how we apply those models in practice and do proof of concept models using synthetic data. So in summary, I suppose we feel we have access to a really unique resource with the epilepsy patient record. We feel we can achieve analytics with impact through this concept of the learning health system, and we can do it in an ethically sound way. So many thanks for your attention.
Thank you very much, Derek. That was really interesting. You know what caught my ear actually was the concept of the synthetic data set. Maybe you could tell us, do you think this is the future of working with personal clinical, like people's data in the future, that you get to work away in the background on this synthetic or, like not someone's personally identifiable data, and then being able to apply it once you have full ethical procedures followed?
I think so. I mean, it's a really rich research area at the moment, and a lot of people are interested in it. Obviously, in terms of, I suppose, the ethical landscape in Ireland, that's evolved a lot even over just the last 2 years with things like the health research regulations, the health consent, research consent declarations committee, the National Ethics Committee. So I suppose we have to navigate those issues, and it's still evolving. So in the absence of while those things are still been put in place, I think the use of things like synthetic data really give us a way of prototyping and developing our models while still preserving patient privacy and doing things in the right way and to move things forward.
Absolutely, as it should be. Thank you very much, Derek. And we'll move on to our third speaker, Professor Claire Gormley. And Claire is in the UCD School of Mathematics and Statistics, and she's part of the Insight Centre. Claire is a methodological statistician with an applied focus. She develops bespoke statistical methods to solve complex data. And with these methods, researchers actually solve applied problems. And this is where it gets interesting across a range of disciplines, including epigenetics, metabolomics, genomics, social science, and also sports science and political science, which I hadn't even considered as an area you could apply it to. So I'm looking forward to Claire's talk on joint modelling of anthropometric biochemical data and genetic data. Claire, I will let you take the stage.
Great, thanks very much, Marie. So yeah, as introduced, I'm definitely coming at this today from the data science side of this intersection with neurology and within the world of data science within the field of statistics. So next slide, please, Richard, thanks. So it was my interest in this research area and in any research, applied area of research really, is that it helps motivate a lot of the research in the field of statistics. So if you think of let's take some of the epilepsy patients that were referred to in the previous talks, from those we record lots of data. So it's really rich, it's very complex, it's interlinked, it's of all shapes and forms. So it could be biochemical data like the VOCs, for example, that Alan mentioned, genomic data, which is just huge in terms of its dimension. We could have images, you could have the streaming data, again, that was alluded to. So we have lots of statistical tools. Most people probably have heard of a t-test once upon a time, but we can't use a t-test for every type of data. So there's different tools for different types of data. And then the question is, well, which is the right tool to use? So our task as statisticians is to develop these statistical methods that we can use to analyze these different data types to help us in this context understand, how a disease works or the mechanisms within it, and importantly to inform treatment plans. And in particular, I think this is where statistics can contribute to this intersection in particular, is in the aspect of personalized treatment decisions. So my own slightly selfish view is that I like to view all of these data in the one go. So rather than having a tool to apply to imaging data and a tool to apply to genomic data, is to try and integrate these data sets and learn from all of them in a contiguous or simultaneous way. Next slide, please, Richard. Thanks. So this is just an example of some work did a few years ago with researchers in the Conway Institute in University College Dublin, where they came to us with a set of data on a group of study participants, and they wanted to stratify that group of participants into separate cohorts. So you could imagine, let's say you have a set of epileptic patients and you're trying to see are there different sets of those patients that are similar to each other. So the data that we had was both like anthropometric, so things like you can see there on the left side of the slide, waist circumference and body mass index, things like we would all know. Also biochemical data, so things like different fatty acid levels that were in the patient's blood, but then also this swathe of genetic data, so in the form of what's called SNP data. So you'll hear a little in the next talk about other types of genetic data, but this was single nucleotide polymorphism data. So our task was, rather than look at the anthropometric data and find these cohorts and then look at the biochemical data and find the cohorts and then look at the genetic data and find the cohorts, to try and do that in one contiguous way. So we had to draw on statistical tools to allow us to do that, and the plot here sort of shows what we uncovered. So you can see we uncovered that there were two cohorts in the set. And for example, if you look at the BMI row, you can see the green cohort was a group of participants that had a much lower BMI, so farther down the axis than the red blob alongside it there, which was cluster 2. So we allowed to dig into the patients that are within these cohorts and understand what their differences are and what separates them. Interestingly then, equally, we get to sharpen the microscope on the genetic data, and we can dig in then and say, okay, well, what are the genetics that are causing some or not are causing are different between these two cohorts. And for example, this is just an example of one SNP on the CETP gene, and you can see in one cluster there was 52% of that cluster had the GG version of this SNP, whereas in cluster 2 predominantly it was the heterozygous gene, the 57% of those patients. So then you can zoom in on genes in certain regions and try and understand what's happening. Next slide, please, Richard, thanks. But I think a really important piece, and I think again this really speaks to the application area here, is being conscious of the uncertainty in these things. Data are real, but they're not perfect, and we always have to worry about the uncertainty that we may be that is in the inference and that what we make from them. So this is an example of an output that we can get from these statistical models that worry about this uncertainty, where along the bottom we have each of the 500 people in the study, and on the vertical lines are basically how uncertain we are about which cohort they belong to. So if I'm a patient and the red cluster are getting a horrible drug and the green cluster are getting a much nicer drug with less side effects, I'd like to be pretty certain that I really need to be in that red cluster. So most of the people here have a cluster uncertainty down near 0 along the bottom. They're fine. We're very certain you're in there. But if you're one of these patients that has a very high uncertainty, this is a point where you'd like to intervene and say, right, well, maybe we need to look at a slightly more personalized plan here. And then final slide, please, Richard, thanks. And then just a totally different application, but again, I felt it might have resonance with the neurology field, is again some recent work with Professor Lorraine Brennan in the Conway Institute in UCD, where she develops metabolomic biomarkers for food. So would hear a lot about biomarkers and then how we can relate them to a trait of interest. But the unique piece here was that she had multiple biomarkers, metabolomic biomarkers, just from a urine sample, 4 in particular. And from those 4 markers, can we infer how much, in this case, apple each of the participants had consumed? So again, you can imagine analogies in the neurology world where you have biomarkers and you're trying to infer a trait. And here we have to build, here we used a Bayesian statistical model, again, that have to be bespoke and opposite to the data at hand. And again, that gives us uncertainty. So here are examples of 2 people in the study, observation 82 and 13 for privacy reasons. And you can see that the plots give us sort of an idea of how much apple they have consumed. So for observation 82, you can see that, you know, we're pretty sure they've eaten around 300 grams of apple. But for observation 13, we need to do a little bit more work. Looks like they might have eaten about 40 grams. They may have eaten about 100 grams. You can see we sort of have two humps in our plot here. So, you know, speaking to this piece about the individualized nature of our inference and the uncertainty that we can bring to that by using statistical methods. So I think there's a really rich opportunity at this intersection, and a lot that statistical methods can give, but equally our work is inspired by these real application areas. So look forward to the exciting research that will happen at this interface in NeuroInsight.
That's fantastic. Thank you so much, Claire. I was just looking at that and going, oh, an apple a day keeps the statistician at play.
Very good.
or maybe 100 grams of apple a day. Thank you very much for that. And so we've heard a lot about how data science is actually helping advance different areas in health. But what about if we flip it and look at statistics and ask, are there any grand challenges left in your field that you feel that working with all this health data is actually helping advance it for you or open up new areas?
Oh, absolutely, and that's in a sense, you know, the great thing about the field of statistics is that two-way street. You know, we very much, you know, I never would have sat down and said, right, well, I'm going to develop a method that can analyze 4 things and predict 1 thing from it, you know, without the applied people coming and saying, here's our problem, and then that gives us ideas to push the boundaries in our own discipline to develop and invent those methods that aren't in existence at present. So particularly in the area of this mixed data, where you have different sources coming from different areas, and the scalability, I suppose, is the really niche piece in that as well. You know, as the scale of these data that are available grows, while often number of patients remains small. So you have a small sort of set of people, but you have huge data on them. And I think that's a place where statistics, you know, has great scope for development, is in developing models to tackle that balance of small observations, but huge data. And that can be inspired and driven on by these real application areas. And it's great to see sort of the real impact then, you know, in, you know, our somewhat, you know, slightly abstract work translating down into, you know, actual problems and helping solve real problems.
Absolutely, it's exciting stuff. Thank you very much, Claire. And now we're going to move on to our speaker number 4, Professor Deirdre Kilbane. Deirdre is head of the division of the Emerging Networks Laboratory in the Walton Institute and the Waterford Institute of Technology, and she's also part of Future Neuro. And Deirdre's research combines nanotech, or AI, quantum communication, and this is to develop innovative technologies for the agriculture, healthcare, and ICT sectors. There are so many exciting buzzwords in that. I love it. Nanotech and AI and quantum communication. I'm really looking forward to Deirdre's talk. Her lightning talk is about using AI to understand ncRNAs, which are biomarkers for neurological disorders. And I will give the stage to you now, Deirdre.
Thanks very much, Marie. I'm delighted to be here today to tell you about our research ideas. And the research has come out of the Project Prime, which is a European FET open project that I coordinate. And my collaborators are from Future Neuro and the Royal College of Surgeons, Professor David Henschel and Professor Jochen Prenn. So the aim of the Prime project is to develop a living brain implant that can sense and suppress epilepsy seizures before they happen. And epilepsy is one of the most common neurological disorders that affects about 1% of the population, or 60 million people globally. So for the people who suffer from epilepsy, there are certain treatments and drugs available, but they aren't always very effective. And for severe cases, the patient can suffer from a large number of seizures during the day, and this makes independent living very difficult. You can move on to the next slide now, please, Richard. So some of the solutions came from discoveries that were made in the Royal College of Surgeons. Marion Hogg, who is a member of Professor Jochen Prenn's group, measured the blood serum from patients with epilepsy before and after seizures occurred. And she found out that there was an increased amount of tRNA fragments in the blood samples from before a seizure occurring. And this is because the tRNA molecules were becoming very stressed, and then they broke into fragments. And then there was an increased amount of these in the blood. And this could be used as a biomarker for epilepsy. Now, the project that we're interested in doing today, as part of NeuroInsight, is to develop some artificial intelligence tools that can help us to identify more biomarkers for epilepsy and for other neurodegenerative diseases. So this was inspired really by the successful application and powerful application of deep learning algorithms to next generation sequencing, which was really powerful tool for identifying or for finding patterns in these really large data sets. Now, it's really crucial that we can actually identify the transfer RNA molecule structure correctly, because the function of these RNA molecules or transfer RNA molecules is really linked to the structure of the molecule. And we're going to use data sets from experiments and humans in the first instance to test our epilepsy biomarkers. And then after that, if we successfully create these models, these artificial intelligence models, we'd like to expand them then to other neurodegenerative diseases such as ALS, Parkinson's, and Alzheimer's disease. You can move on to the next slide now, please. So we believe this research is hugely beneficial, and it can greatly impact the lives of patients and families of people who suffer from epilepsy. In the first instance, for example, if you look at the patient who suffers from epilepsy, if you can reduce the number of seizures that the patient is experiencing on a daily basis, they can start to live a more independent life. They can consider returning to the workplace or being employed in roles that they couldn't previously have been employed in, more risk roles like becoming pilots, for example. The benefit to the economy is that it alleviates a large amount of stress on the healthcare system and the finance of the healthcare system. Your epilepsy patient now doesn't need to visit hospitals or GPs as often and doesn't need to get treatments from large equipment or expensive equipment. And the last benefit I want to mention is the emotional well-being of the family members of this person who suffers from epilepsy. The carer must always be present to look after a person who's suffering from seizures and must know how to deal with these seizures and also organize the lives of these patients. And so it will be hugely beneficial for the family as a whole.
Thank you very much, Deirdre, for that. It was a really interesting talk. And these kind of AI tools that you're working with and developing, can they be applied to find biomarkers for other diseases, something in addition to epilepsy? Is this something you might be looking at in the future?
So, you know, traditional bioinformatics tools use slightly different models. They use thermodynamic modeling, and they have been used in the past to identify biomarkers for diseases such as cancer or heart disease, and for a number of different diseases. So we would just really like to extend those tools and maybe find more biomarkers or different types of biomarkers, really.
Fantastic. Yeah, it's really a really exciting area altogether. Thank you very much, Deirdre. And we're going to go on to Professor Tomás Ward now. Tomás Ward's a full professor of data analytics at DCU. His expertise is in digital biomarkers, data analytics in health and human performance, neurotechnology, biosignal processing, and wearable sensors. And Tomás's focus is on machine learning applied to physiological time series. And he's part of the Insight Data Center or Center for Data Analytics as well. And he holds the AIB chair in data analytics. So Tomás's lightning talk today is going to be about using AI outside the clinic, enabling better understanding and treatment of neurological conditions.
Thanks, Marie. So I'm interested in the development of AI for improved tracking of human health and behavior outside the lab in real world settings. There's a lot there of various areas that Marie mentioned, but in a nutshell, that's what I'm interested in. And so this research leads us to develop enabling methodologies which can allow us to better understand and treat neurological conditions, I think, in the context of NeuroInsight. And you might ask, why this focus on real world measurement outside the lab? Why is that important? You think the lab, that's where all the serious work is done. Why outside the lab? Well, there are a number of things that motivate us here. The first, and it's shown on this slide, the first motivation is ecological context. And what I mean there is one motivation is to capture relevant health data in context. And we've heard many examples there about seizures in patients with epilepsy, and that is a good example. I'm going to give that same example for ecological context. So, seizure events and their frequency are really important data to understand the efficacy of treatment. And one approach to gather that data is to simply ask the patients when they visit the doctor about the recent seizure history. In that case, of course, we're relying on the patient's memory for this data. We're relying on patient recall. And as we're all aware, our memories may not be perfect, and so such an approach may miss data. So perhaps a better approach is to use some sort of sensor system to automatically detect and record such seizure events in a way that does not rely on the patient, something that's more objective. And this gives us higher fidelity, more reliable data for understanding the condition. So it's a good example of ecological context. So another motivation would be to broaden the net for the discovery of new biomarkers. So through the collection of data in context, through the use of sensors and combining this with other health-related information, we could develop high-quality data sets in which we could look for patterns that might allow us to understand the severity or progression of a disease. So in particular, we were interested in the development of, or in the inclusion of digital data to what some people call digital biomarkers, based on, for example, people's interaction with their phones, their wearables, or other consumer electronics that are available to us. Maybe as an example, imagine using a patient's interaction with their Siri or their Alexa to monitor for neurological symptoms, for example, voice tremor and Parkinson's disease, so use that kind of this other channel in our everyday interactions to kind of develop enhanced biomarkers. And so the final motivation, and there are many motivations, I'm just picking three here. So the final motivation I will mention here is the promise of digital therapeutics. So if we think further into the future and we have a world in which we can monitor patients outside the lab, at home, as they live their everyday lives, imagine how we might recognize triggers or signs of deterioration and then take some sort of intervention. We just saw a good example there from Smeaton on the dogs and the intervention in the context of seizure. But there'd be all sorts. For example, a diabetic patient might, benefit from exercise, and if you can monitor that they're not doing so much exercise or their dietary intake has changed, you might intervene or advise. Anyway, this area is this emerging area is called digital therapeutics, and it spans a wide range of methodologies and application areas. And we're very excited to explore these for any chronic neurological condition. So we've an open mind there, but we have the tools and methodologies to actually deploy and try out ideas there. So we're motivated, that's for sure. Now, standing back, it's clear that all these innovations are reliant on data capture outside the lab in the real world. Now, how do we do that? So thankfully, we have options. So we have from the ubiquitous wearables. So many people have Fitbits, many people, their phones themselves are capturing a lot of data about what we do and our movement and so on. It's all digitized, it's available. And we like to use, or have been using, consumer wearables, off-the-shelf consumer wearables to see what we can do in terms of enhancing data capture. But we go beyond just the everyday common or garden wearables to more exotic technology, such as, for example, brain monitoring the home, or EEG as it's called. So this we can, in the context of NeuroInsight, and this is what NeuroInsight is enabling actually, we have a company partner called Cumulus Neuro, and through Cumulus Neuro, we're able to monitor a person's brainwaves at home. So this is a great means or opportunity to monitor the brain regularly over time, at home, at work, at rest, asleep, at play. So it allows us to see the brain in its natural environment, if you like, okay? So this device that, as I said, is developed by Cumulus Neuro is a really superb innovation because it allows the ease of capture of brain activity by patients themselves. And that's a big deal, because before this, an EEG is something that's carried out by a technician in a very, I suppose, a very formal environment. So this is really empowering. And it's a great step forward in advancing translational research in neuroscience, in my opinion. So these data sources present great opportunities for us to advance, I suppose, clinical research, but they're not without challenges. And that's why we need this sort of interaction here between insight and future neuro. So data analytics then is the key to unlocking this. So it is to data analytics and machine learning that we have to turn to address these challenges of really acquiring high quality data outside the lab in the real world. From making brain signals measured at home easier to acquire, to extracting subtle insights from your Fitbit, we use AI to develop deep value from sensor data. And as an example, just to focus on the brain signal slide I had there, brain signals are really noisy. And from looking at brain signals, if you looked at the electrical signals in the brain, and you turned it to sound, for example, it would sound like a tuned out radio. It just sounds like hiss. You can't see any patterns in it. But from the use of AI, we can see the brain at work. We can see when the brain is paying attention. We can see when the brain is at rest, or when the brain is relaxed, or when the brain is asleep. And so that's what we can do with AI. And so it's this combination of skills in clinical sciences, machine learning, engineering, and computer science that I think we can enable through NeuroInsight, and can allow really exciting future projects to happen. So if you're interested in that space, come talk to us.
Tomas, thank you so much for that. I have so many questions, but I'll stick with one. So you talked about the idea of using consumer wearables or smart home devices that we already all have in our home and we're using every day. Does that mean you foresee that maybe in the next 5 years or so that we could have consumer apps sitting on these wearables and devices that are doing all of these really complex things using the machine learning you're developing now, that it's something that people could be actually, seeing if they'd signs of Parkinson's just from an app on their phone that they downloaded in like under a minute?
Yeah, absolutely. And that sort of measurement in the wild facilitates that, and particularly through open data standards and so on, it allows AI pipeline to be built to pull additional insight from these devices, for sure. But while we can do that measurement in the wild, it's important to note it's not the Wild West. And so another important aspect to what we do is kind of ethical AI. This is people's data. This is people's health related data. So we are we're very keen to very carefully facilitate the development of kind of new measures from these devices, but it has to be done in a very cautious way, in a very careful way, and with the full co-design and co-creation of patients and end users. Not absolutely. Quite the question you asked, but anyway, that's something I wanted to say anyway.
No, absolutely, because I think I'd say the audience are probably thinking on one hand, is that data robust enough to actually diagnose somebody on the run, kind of? Just as
that needs to be done very carefully, and it's an open question, and it's very application dependent. But yeah.
And the second question being how usually your doctor sits down with you, and there's a whole process behind breaking medical news to people, and counseling that goes along with it. So you can't just give them an app alert going, you might have Parkinson's.
Absolutely. Well, again, this is why we need this dialogue between all the stakeholders. We can't let the engineers run riot or the computer scientists run riot. Or what usually happens is the patients run riot. They're Googling left, right, and center, and everyone's convinced they've got everything right. We have to create this sort of a space in which there's this kind of co-creation, co-development activity, and all done. Again, you've seen the ethical AI is incredibly important. I've never seen regulations from the EU now to try and structure that, and that's increasingly important. Because AI is an integral part of many future medical devices, and even regulatory bodies are scratching their heads about how they're going to regulate such kind of disruptive technology that is open to that needs to be carefully deployed. Put it that way.
Absolutely. And before I move on to John Piero, I just want to say, I suppose that's where we can see today that non-expert publics and PPI groups especially come into play. And that's what Science Week is all about, getting the wider public involved. So thank you very much for that, Tomas. And our last speaker, and certainly not least, is Professor John Piero Cavalieri. So John Piero is actually the Deputy Director of Future Neuro, Professor of Human Genetics, and he runs the Human Genetic Variation Research Group at RCSI. And his research group works on diverse topics at the interface of computational biology, clinical research, and human evolution. And his team are applying the latest generation of DNA sequencing techniques to identify novel genetic indicators related to epilepsy across populations. So John Piero's talk is going to be on artificial intelligence as a tool for faster, more accurate genetic diagnosis. So yeah, John Piero, I will let you take the stage now. Thank you very much for providing the last talk of the afternoon.
Thanks, Marie, and it's fantastic to be here today and to hear all these exciting talks and opportunities. So this talk is about genomic diagnostics in our hospital. So the challenge is simply to find the specific genetic cause of an individual's rare disease. And our ability to do that has really been transformed over the last number of years by developments in DNA sequencing technology. So you might be able to see from this image that today we can sequence a full human genome. So that's all 3 billion bases of your DNA for about €1,000. But if you went back to 2003, when I think it was Clinton and Tony Blair announced the first sequencing of a human genome, that effort cost tens or hundreds of millions of euro and took thousands of people many, many years to deliver. So it's much, much cheaper and easier to generate a lot of genetic sequence today. And that means, in theory, that we can start to integrate this technology into our hospitals and our diagnostic systems. But, Richard, if you can progress there one. But you might anticipate that the analysis of a large data set with 3 billion elements in it is quite a large computational lift. And indeed, if you sequence the genomes of any two individuals and compare them to each other, you'll see that they differ at about 0.1% of their genome. So it doesn't sound like a lot, but 0.1% of 3 billion equals 3 million points. So say if an individual has a rare disease that's caused by a specific mutation, it's a bit of a needle in a haystack effort to be able to filter through those 3 million differences to find the one that is causing their disease. And this is where we're hoping that AI can help. So Richard, if you can go forward one. So if we take a closer look at the process of genomic diagnostics, it begins with the patient presenting at the clinic and say where the clinician suspects a rare disease, maybe a rare form of particular neurological condition. They will take a blood sample, they'll send it to the lab will extract DNA, they'll use some of that sequencing technology to sequence those 3 billion bases of the individual's DNA. And one of the first computationally heavy tasks is to identify the 3 million bases where they differ from another individual. And we call this process variant calling. So what are the points that you differ from someone else? And then having called all those 3 million variants, the next step is to annotate them. So what do we mean by annotation? That's basically determining, does each variant fall into a gene, number one? If it does, what gene is it? What is it doing to that gene? Is it changing the sequence of the amino acids in the protein? And also, very importantly, how rare is it in the population? You remember Claire's slide where she showed a SNP, which is one of those genetic variants. She referred to it as 50% in one patient group. So that'd be a really high frequency variant. For this process of finding a clear genetic cause, the variants tend to be extremely rare in a population. And that's why the diseases in question are rare. So once you've annotated all the 3 million variants, you then need to shortlist them. So that's shortlisting the 3 million to maybe 30 or 40 variants that are present in genes known to cause that disease. So maybe there's 100 different genes that are known to cause epilepsy. If the sequencing is for epilepsy, you would shortlist the variants in those genes, and also to the variants that are very, very rare in the population and predicted to be damaging. That shortlist would be pushed forward to a multidisciplinary team meeting where each one of the 30 would be interpreted by a clinical geneticist, the bioinformatics team that undertook all those previous steps, and also the referring clinician. And what happens there is they pull additional information out of the clinical record to help interrogate each particular variant to determine which one or combination perhaps might be pathogenic and disease causing, and from that a report is produced. So this pipeline is something that in Ireland here, future neuro researchers have helped realize and integrate into our neurology clinics. So we like to think in a small way, but a significant way, this is part of creating and realizing our future. But it could be improved, in particular through the application of AI. So just thinking that through, if you look at the variant calling step, there are AI algorithms now that are based on these things called convoluted neural networks that are able to better identify the points where you differ from someone else, those 3 million points. Because if you don't get that calling right, you end up with lots of what we call false positives in the sequence, where a mutation is called, but actually it's not a real one that the patient carries. And you can imagine if you carry that through the process, it'll cause all sorts of distractions. So a nice example of that type of AI algorithm, there's one called deep variant, but these things can be improved. The next step that AI can potentially help is in annotation. So for example, again, you can use convoluted neural networks to look at particular patterns of protein domains, to see which domains are more important or conserved across primates, because conservation is indicative of importance, especially when it comes to mutations. So primate AI is a nice example of that. And the final point where you could integrate AI, or AI has been integrated to date, is around shortlisting of variants. So there's a really cool algorithm called DeepC that looks beyond the coding part of the genome. Remember, only 3% of your genome actually codes for genes. 97% is sometimes referred to as the dark matter of the genome. So algorithms like DeepC are able to infer maybe transcription binding sites or histone markers, things like that, that when they're mutated could be contributing to disease. So the opportunity, I think that's a really exciting one for NeuroInsight, is to create projects around this pipeline to see how better can we integrate AI into the genomic diagnostic process, or perhaps are there additional algorithms that we could create to integrate here, and doing so will lead to a faster, more effective, more impactful diagnosis for people living with neurological conditions in Ireland and abroad. So thanks, Mary, and I'll stop there.
Thank you very much, Jean-Pierre. That was fascinating. And one thing that I think maybe others will be curious about, which I am, is where does this data come from for your work? Where do you get it? And I suppose mirroring the question to Tomas as well, what are the ethical considerations of working with such genetic data?
Yeah, so in this example, like if you look on the cartoon here, there's a little blood sample. So the data here is coming from the patient. So they would have consented to having a genetic test done. And in fact, there's a very important counseling step that I didn't go into at all in this process. But the data here would come from the patient. But a really important point, to understand the relevance of genetic variation that occurs within an individual, you need to be able to study it in the context of hundreds of thousands of other genomes that have been sequenced globally, right? So there's a lot of genetic variation, like points that we differ in our genome, that have been placed in databases. So they don't necessarily put the whole sequence of an individual into a database. It would be the particular points that individuals differ. And this would all be anonymized data in the form that's in the databases. But having those databases present, and they're indicated here by the little white or the yellow tubes, by the way, having that context present is really important to interpret and understand what variants have been seen previously, how frequent are they, and critically, have they been seen in that condition before? Like today, we understand a decent percentage of the mutations that can cause cystic fibrosis, that can cause rare forms of epilepsy, Huntington's disease, many, many other conditions and neurological disorders, because the disease-causing mutations have been put into databases, so that if we sequence in Dublin, patient X in this diagram, you can understand whether it's been seen before, in what condition, how frequent, or is it only observed in so-called healthy individuals. So the context of sharing data and information is really, really critical for us as individuals and as a population to get the maximum benefit for society out of genomic data. But obviously there are consent issues that need to be addressed. It needs to be done in a very, very ethically secure and sustainable environment to enable this to happen at the scale that we need it to, if we want to get the maximum benefit from it.
Fantastic. Thank you very much, Gian Piero. And thank you to all of our panelists. It was really a fascinating lineup. I think everybody got a little something from that, and we're dying to know, you know, what NeuroInsight will come up with in the future. And for everyone who's attended here today, just to let you know, if you want to find out what NeuroInsight is up to, follow updates on the NeuroInsight Twitter account. It's just at NeuroInsight EU. So here it is on screen. And also, yeah, go head along to the website. And thank you very much, Richard, for organising today's webcast as well. And thank you, everyone, of course, for taking your lunchtime to come along to the talk. Hope you got to eat your sambos while you were listening in. Thank you very much again, everyone.
Thanks, Marie. Thanks, Marie, for hosting. And yeah, sorry, we've gone a few minutes over time, so we rushed through the questions at the end, but I think they're all answered in the chat where people asked them. So yeah, hope everyone had a good time. Thanks a lot.